So I'm recording now, just so you know, uh, and we'll post this recording to the FAANG website as we did for the last session. Um, I see a couple more people are coming in. We're almost at 30. We are at 30. That's great. Um, Without further ado, we have four speakers this morning. They've been asked to keep their talks to about 12 minutes to allow several minutes for questions. And then we hope to have a few minutes at the end to just discuss with everyone that's interested, how should the task force prepare for the next year, especially the next session that we'll have in the fall, that's scheduled for the fall. So the first speaker is Fernando Baez from University of Vermont. And I think, Fernando, if you're ready, I think we can just go ahead and get started. I'll continue to add people as they're coming in.
But we can't hear you, Fernando.
Yeah, you're still muted.
Can you see these slides correctly, Chris?
They're presenter mode right now.
Of course. Space.
There you go. I can see the slides and hear you.
Great. Thank you so much, Chris. My name is Fernando, and I'm an associate professor here at Virginia Tech, and really appreciate the opportunity to present some of our work and that recent work in single cell that we have started about a year or so ago. I will try to maintain everything super brief and quick, and I would love to take any questions that you might have later on. So this is going to be a little bit of the outline that we're going to talk about this morning. I always like to start with presenting the model that we work with. Essentially, we work in genomics of the embryo development with a heavy focus on understanding why embryos survive, or otherwise they halt their development or arrest their development and terminate a potential pregnancy. We have a system to produce those embryos in vitro here. And essentially, this is the focal point of our program and has been for a while. So on this slide, you're seeing some images of some embryos as the development progress. It starts, I will not go through with details given the time, but it starts with the one cell embryo on day one. It goes all the way throughout cell progression and mitosis. And you're going to see some differentiation of cell functionality at some time at this point right here when there's a compaction and moral formation, all the way to when the embryo starts a cavitation, which you're seeing here, and there's a definite formation of different cell types. Here you're seeing the inner cell mass, which will form the fetus proper, and then you're going to see some cell layer here, which we call trophectoderm, which will then form the placenta of the embryo. So this is our model that we work with. These are some images of embryos that develop past day 7 or 8. These are day 12 in vitro embryos, and you can see that they can get quite big. If you think about this metric here being about half a millimeter in distance. So they can get quite big. And what you're seeing on this slide is something like this. The same embryo you have on this image, and we have essentially 4 cell types on these embryos. You have the epiblast right here, which will form the fetus proper. You have a layer of cells surrounding the embryo, which will be the trophoblast, again forming the placenta. And then you have an inner layer of cells circumventing the trophoblast, but from the inside of this cavity, which is the hypoblast. I do not have other cell types here, but they can be identified. They're called mesoderm. And those cell types, they can be identified with specific markers. Here you have immunostaining of some specific markers. For instance, for epiblast, as you can see on this grouping of cells, which resemble this area right here, hypoblast, the inner layer of the cells of the cavity, the trophoblast or trophectoderm, the outer layer of the cavity, and these are the major three cell types that you find in nearly every embryo. And then sometimes you're going to have mesoderm finding in some embryos as the development progress. Interestingly enough, we can benefit from the fact that understanding the embryology of this process has implications for livestock species, such as cows, as well as humans, because you can see on this image here that there's a great deal of resemblance of the embryo development in cows, for instance, in humans. Both species progress with development with the hypoblast, inner cell mass, and trophectoderm, including further into the development where there are minor differences only on the morphological aspect, and the functional aspect of those cells are super similar. So as I mentioned before, we have entered this world of single cell biology about a year or so ago. And we wanted to start by working with these embryos. And one thing that we have been working on in our program is to try and profile the RNA transcriptome of single embryos. So whenever possible, we isolate the embryos and study them as a single individual. Naturally, we can break this embryo apart. As you can see here, the solution of the single cells into little droplets in the media. But it turns out we have always been under a limitation of the number of cells that one can work with if you're going to go to microfluidics. As you can see here, the microfluidics wouldn't pretty much not allow us to work with those embryos at the single embryo level. Hence, we moved forward to working with a microfluidics-free approach, which is the PIP-seq. And this is just a diagram showing how that PIP-seq works. I won't get into the details given the time constraint, but the main point here is that you have a tube where the solutions and some other barcoded embedded reagents are present in a tube, and then we can drop our cells in there, and through emulsification, each cell will be captured in this emulsified solution containing the barcodes and all the reagents necessary to progress. Literally, all you need is a vortex, a specific vortex, and a thermocycler to make this work. No need for fancy microfluidics. So we progressed with that, and here the publication associated with that. We progressed with that approach. Fluid Biosciences does have a pipeline for us to work with this data set that they produce. So there's a companion pipeline associated with that. And this is just an idea or a very brief observation of some of the data sets that we have produced. Each one of these samples is one embryo, and you can see the number of genes observed and number of cells captured out of each embryo. This is very much in line of what we would have. I can tell you that I have 2 days of development in here, and that's pretty much in line of what we would expect. Some members are bigger than others, and that's not so surprising that we have a different number of cells captured from individuals. Naturally, we have to put these guys through some quality control, limiting the number, establishing a minimal number of reads that we would require, and move forward. In this case, in this work, we've moved forward with 5 samples to progress with our analysis. Naturally, the pipeline, there are extensive guidelines for pipelines when you work with single cell analysis, right? You can read as much as you want, and you're going to always have default parameters to work with, but you do have some types of benchmarking of particularly cell integration of their data which will show that you have a variety of possibilities of pipelines, approaches, methods to work with when it comes to putting this data together. Because if you think about it, all these 5 samples produced, each one produced one data set. The question becomes now, which pipeline do I use? Which integration do I use? And how do I define clusters? I did go around asking people, and most of the answers that I got were not to my satisfaction. So I decided to put my data set through my own pipeline of benchmarking. So essentially, when I put all the cells, merging them together, put them through a transformation, I put this data set through a series of possibilities of pipelines that you can identify with different integration methods, different parameters, and so on and so forth, including the graph formation of clustering. And we essentially annotated cells automatically using specific canonical markers. We did evaluate that based on some clustering associated with cell numbers and markers identified, and ended up taking a look at what's the biological outcome out of that. The one thing that we noticed up front, and I will keep this short, but the one thing we noticed up front is that some methods for integration result in an enormous amount of cells that are not annotated. For whatever reason, they just cannot be annotated. For instance, Harmony and RPCA integrations result in the most number of cells that are not annotated for whatever reason. Other methods do annotate cells quite well. Interestingly enough, there are cells that you find less stable in terms of the identification. For instance, here you have the number of cells identified from epiblast type. You have 0 cell identification all the way to some 200, 400 cells. Mesoderm, 0 to 400 cells identified. Other cell types are a little bit more stable, probably because of their transcriptome profiling, some including the hypoblast cells, for instance, whereas some particular methods do have a wide variety of numbers of cells identified. Moving forward, with all that data, we kind of clustered them, and this is kind of the clustering that you can identify. A few things jumped to our eyes here. For instance, some groups of integration or combination of parameters resulted in absolutely no or zero or a minimal identification of epiblast cells. Now, I told you and I showed you that I do know that epiblast cells are part of our system. They are in our embryos. We can visualize them. And I can tell you that we were very carefully when breaking those embryos apart to make sure that those cells were in our samples. So that kind of raises a red flag. Now, if we move to the biology, so I will keep us on time, I will show you just the results of the biological insights obtained from one cell type, in this case, the trophoblast cells. What you can see is that when I do a gene ontology analysis out of particular integration methods or combinations, and those are the integration that you can see here, obtained from different clusters. Like I mentioned a few slides ago, I had 3 clusters, 1, 2, and 3, and I grabbed some of those integrations out of those clusters. You have some integration, some parameters that do get similar outcomes, and you can quantify that similarity, for instance, using Jacquard similarity. But you do notice that you do have a series of biological categories that show or are absent on the parameters depending on how you look at the data. Hence, you will have resulting biological distinct results depending on the type of integration and workflow that you progress with. To keep me on time, here are the take-home messages. Absolutely, integration method and parameter will have an impact on cell clustering, cell identification, marker identified, biological interpretation, which is the most important. And if I can draw from something that I learned from my mentor years ago, Harris Lewing, you got to know your biological system if you want to work with those single cell analysis. You need to know the biology. And I will absolutely conclude with my team who helped me with this, and of course the funding. And I am happy to take questions after when possible.
Thanks,
Thank you.
Fernando.
Thanks, Luis.
Are there any quick questions? Maybe just one quick one, Fernando. So since you see such differences
Yes.
in both like number of cells that are unannotated and some of the results with the annotated cells, especially disturbing is not finding epiblast cells, right?
Yeah.
d- are there intrinsic ways you can test which of the integrations or which of your processes are more likely to be correct? Have you looked into that?
Well, this has become my most critical question, right? Which ones are correct? Because it's very easy to say those that are not correct. What I have been looking at so far to this point is which ones are the most stable and resemble the biology the best. I don't think there's going to be one approach or one combination that is the best, but there might be a few parameters that kind of are congruent to giving me the most appropriate answer. That would be my take on this. But definitely you can isolate some that should not use.
Okay. Thanks. Yeah, I mean, I guess it's hard. We obviously we want to embed it in the literature, but it's difficult. Okay. I
Thank
think in the interest of time, we'll move on. So the next speaker is Alex Klepp from our CRAG from Spain. So Alex, are you there?
Yes, I'm there. I try to
Okay.
share, but I think it's one participant can share at a time.
Yeah, I think you should be able
So,
to share now.
okay, share, right. I think it's this one.
Yep. Starting.
Let me see. Okay, okay, yeah, allow. Right. And let me know if you can see.
Um,
Um, is it correct? You see it well?
no, I see you trying to go to slideshow, but it stays in the slide present, the slide working. There it goes. Yep.
There you go, sir. Okay,
Yep.
okay.
Yep.
So,
That's
well,
good.
so hi, thanks everyone. I present a couple of studies that we are doing in which we combine genetic variation and single cell sequencing analysis with the aim to find variants with a strong functional and causative potential to affect different sperm traits in pigs. Basically, in this slide is what we show. We see here that we apply single nuclei multiome RNA and attack using 10x technologies of the testis. This technology allows us to evaluate gene expression and of the very same cells, open chromatin with attack-seq, open chromatin that is well associated with active regulatory elements that regulate the expression of the genes in this cell. So we can identify gene expression and the regulatory elements that putatively regulate those genes. And then we combine genetic variation with different approaches that you will see in the two examples for sperm traits to identify variants that have a strong causative potential. I wanted first to, let's see if I can, here. The testicle, sorry, here. The testicle contains a complex set of cells well defined from immune cells, endothelial epithelial cells, and among them we have Leydig cells that produce testosterone, important for spermatogenesis, Sertoli cells that produce nutrients and signaling to the germline cells throughout the spermatogenesis. And here in this box, the spermatogenesis cell types from spermatogonia, which proliferate until they start differentiating as spermatocytes which undergo meiosis, the different paternal and maternal genomes recombine, and the resulting cell out of meiosis is spermatids. They are haploid, they contain only one copy of the genome. They are non-spermatids and late spermatids, and they undergo what is called spermiogenesis, in which the sperm cell itself will acquire the morphology and the physiological functionality that we know as a sperm cell. Okay. I present two studies, as I said. One of them is on non-obstructive azoospermia. Some months ago, a company contacted us. They were concerned because they have a close population of boars from artificial insemination centers that 7 years ago, more or less, started showing few animals with azoospermia. Azoospermia is defined as the lack of spermatozoa in the ejaculate, and is non-obstructive because it's not on the conducts but it's on the testicle itself. So it's a spermatogenic failure. So it's in the testes. After discussing with them, I concluded, or I hypothesized that the most likely scenario, simplistic, less expensive to make the experiment, was a recessive model in which is a novel mutation because the population is close. It's a novel mutation not present in devious SNP, in which the animals that are homozygous for this novel mutation are azoospermic, and then the animals that are not azoospermic are either heterozygous or homozygous for the wild type allele. Of course, a father of azoospermic animals has to be heterozygous because he has to transmit the mutation to the offspring, but he himself is fertile. Okay. Under this hypothesis, we sequence the genomes of 5 cases, 1 father and 3 controls, and look for variants fulfilling those conditions. We identified a bit more than 5,000 variants, and interestingly, 4,924 of those variants map to a 27 megabase per region in chromosome 7 with a very high density of variants. There is 212 variants every megabase per. It's a high density of variants. So clearly, this is the candidate haplotype, identical by descent. 27 megabase per region in chromosome 7. Of all those variants, 6 SNPs and 1 short indel were predicted to affect the protein sequence severely of 6 candidate genes, and interestingly, all those genes, this is not transcriptomics or epigenomics, this is whole genome sequencing only. All those 6 genes have strong links to spermatogenesis and azoospermia. We then moved to the single cell, single nuclei multi-omic analysis. We did 1 azoospermic animal and 6 control boars. Here we saw the UMAP. This is basically a clustering method and with visualization in which different cell clusters are clustered according to how similar they are in gene expression. And we can annotate them as peritubular, Sertoli, spermatogenic cell clusters here in the continuum. And once we have got this UMAP, we then look at the proportion of cells that each sample contributed to every cluster. In the first column in this table, we show the annotation. What cell type is this? Spermatogonia, spermatocyte, spermatid, Sertoli, and so on. And then here, this column in red shows the number of cells that the azoospermic sample, labeled as A, contribute to the cluster. And then all the other cells, the healthy cells. And in the last column, we can see the deviation that we observe in the contribution of cells from the azoospermic sample to the cluster. So the larger number is an increase on the expected and lower a decrease on the expected number. And we highlight in yellow those strong deviations above 1.5 or below 0.7. We can see an increase in differentiated spermatogonia, whilst a decrease on spermatocytes and early spermatids, like indicating that something is preventing spermatogonia to differentiate into spermatocytes and into meiosis. This is pretty robust, it seems, according to this data. And then we see an increase on Sertoli cells, on Leydig and peritubular cells, to the point that cluster 11 on Leydig is mainly made exclusively, almost exclusively, of azoospermic cells. So there is clearly distinctive cluster in Leydig, in transcriptome or epigenome or characteristics in these Leydig cells on the azoospermic sample. We also looked at the, sorry, what's here? Okay. We also looked at the gene expression and attack itself, and from the 5,000 variants in the candidate region, the 5,000 candidate variants, we look at how many of them map in attack peaks. 198 do that. We also look at genes that show outlier expression, as shown here in the zoospermic sample when compared to the control samples, here 6. And we use a z-score equal to 3. Looking at this, we found 87 genes mapping in whereas zoospermic region in chromosome 7 that have a clearly outlier expression in the zoospermic sample. And now we intersected positions and we look for variants in attack peaks that map less than 10 kb or even within outlier genes in the zoospermic samples. And we found 58 variants, all of them SNPs that map nearby or within 23 outlier genes, 7 of which have clear links, known links to zoospermia. So we consider, and of course this is ongoing work, is a prioritization only, but that the 58 variants, potentially regulatory variants, and the 7 potentially altering protein sequence are strong candidates to be the zoospermic variant. The other study we want to show how we combine genetic variation and single-cell analysis is to identify variants with a strong potential to affect spermiogenesis. Remember that spermiogenesis involves spermatids, which are haploid cells. Under the hypothesis that the variants that affect spermiogenesis will alter the number of spermatids, we can assume that in heterozygous sites, we can compare the number of cells carrying each allele, the number of spermatids carrying each allele, and those variants in which the number of cells carrying each allele is not equal, they deviate from the expected ratio of 0.5, can be presumed to affect spermiogenesis or at least spermatid development. The first thing we did is whole genome sequencing of the diploid genome of 4 boars.
Alex, just 1 minute.
Yep.
1 minute.
1 minute, okay. Okay, heterozygous sites in the that were robust heterozygous sites in this board. Then we carried single-cell multiome analysis, attack and RNA, and identified in the pseudobulk, so at the gene expression level or attack level, in the spermatid cluster from every sample, those variants that were heterozygous positions in which there would be a allelic ratio distortion in the ratio of each allele. And for those, and we call it ARI-read, for those that showed ARI-read, we genotyped the cells. Few of them could be genotyped, but for those that could be genotyped at the cell level, we evaluated the number of cells that carry differences, different cells carry one allele or the other, okay, with a feature exact test. Here we show a cluster with the spermatids, 3,600 spermatids, and at the pseudobulk level, we can see some clear clusters in chromosome 6 near the EA1 gene, and for the attack in the same location, and also near the ROPN1L gene. All those genes are linked to zoospermia. And when we look at the ARI cell level, of course, the number of variants is less, but interestingly, what we see here is that most of the ARI-read SNPs that could be genotyping at the cell level also showed ARI cell, and that the ARI cell and ARI-read, it goes always in the same direction with strong correlations, indicating that ARI cell is ubiquitous and present. And there are several genes that are displaying ARI cell and that have strong links to spermatogenesis. Of course, I have collaborators, PhD students working on that, and funding that I would like to acknowledge, and I take your questions. Thanks.
Thanks so much, Alex. That's very interesting work, combining the genetics and the single cell. So there's a question in the chat. Did you genotype spermatid cells using multi-ohm data?
Yes, so what we do is first we identify heterozygous sites with whole genome sequencing, which is way more robust. We trust those heterozygous sites, and then we look at the spermatid data from the single cell data, RNA and attack, and we try to genotype all those variants. Of course, many variants fail to be genotyped, but those can be genotyped. We are ready to evaluate the allele representation imbalance.
So a combination of the RNA-seq data as well as the attack-seq data to genotype. Okay.
Yes. Yes.
Any other quick questions? If not, thanks very much, Alex. Interesting story. So let's go on to the next
Thank you.
speaker, Hervé Acloc from INRA in France.
A ver.
So Alex is going to
Yep.
share.
Yep, if I can share, let me see how to do that. What is it? Sorry, I don't know what's going on. 1 second. 1 second, if I manage. Look, I will log out and log in again because I'm failing here, okay? Sorry.
Okay. Okay, he might be gone now. Go ahead, Hervé.
You can see my screen.
Yep.
Okay, so good morning, good afternoon, everyone. So I will just present some results we got from single cell datasets working on pig blastocyst development. So coming back to the presentation of Fernando, we are mostly interested in the different embryonic stages that differ from human and mice in ungulates, like in pigs and cows, where the blastocyst is staying in the uterus for many days and will develop with a strong development of extraembryonic tissues, like you can see here in purple and light blue. But the pluripotent stem cells that are here in red will give rise to the fetus, but do not change so much. And we wanted to know a bit better what were the gene regulatory networks at play in these different cell types, to better characterize also the subpopulations that we can observe in the different tissues. And also, if we can see some key cell-cell interactions between either extraembryonic and embryonic cells. So for that, we worked with the 10x genomics technology at different stages. So we have done single-cell RNA-seq only from the blastocyst at E5 to E11. And we have been able to do single-cell multiomics, so at the same time, ATAC-seq and RNA-seq in the same cell, from E7 to E11. And the main problem not to do it at E5 is that we were not able to get enough cells from the embryos. So after that, for the single-cell RNA-seq, it's quite standard process. I will not develop here because it's widely used now for many labs. But I will just present some important results. So what we saw first is that there is a clear dichotomy between early stages that are common to all mammals and the ones that we can see later on, so from E7 to E11. And we can see from our data sets that something is missing between the cells that are here in green and the other one in red. And it will be necessary to come back to the sampling to be able to get the cells that are going to do the connection between this window and this window. The second things we have done is to come back to identifying specific subpopulations. So this is for the trophectoderm. So the trophectoderm will give rise to the placenta, and we observe that we have some specific subpopulation expressing EL1 beta. And EL1 beta is important for the elongation and the implantation of the embryo, but we can see here that this is a specific subpopulation, and it's only distributed at these stages around the embryonic disc here. So it's not only diffuse, but it's only specific area of the embryos. The second point we observe is the new potential stem cell populations in the trophoblast, and also the one that is going to enter apoptosis and maybe the rubber layer here that covers the embryo at the beginning and will disappear after. After that, we were more interested in identifying in this subpopulation specific gene regulatory networks, and for that we use the SENIC algorithm developed by the groups of Hertz in Belgium. And the idea is to link transcription factor to their direct target by looking at a specific motif of this transcription factor in the promoter of the targets. But when you do that with a single-cell RNA-seq data, you will see that you will get a lot of transcription factors and irregulars, and it's difficult to really filter what is of interest, what is a false positive one and a false positive one. So we try to refine this by comparing with different other data sets. So we use some from humans single cell data sets and other from the pigs. And you can see in this case that you can start to get more interesting information, either by getting transcription factors that are conserved between the two species, or at least between two studies on pigs. But the nice thing is that we were able to do single cell omics, and we tried to add this information to refine our regulance study. So first, we confirm that we can get different subpopulation of the embryo, the three main subpopulation. And here you have the expression and the accessibility. And after that, we use the other algorithm from the group of Hertz in Belgium, named Scenic Plus, because this one is based on the same process idea of Scenic, but you're adding the fact that the motif should be inaccessible chromatin, a DNA, new DNA. And in this case, you can really get more accurate results. And you can see here that in the pig embryo, in the epiblast, you have many transcription factors that are conserved and active during times. And you don't have much differences between early embryos or late ones. But when you look at extra-embryonic tissues, like for the trophectoderm here, you can see a dynamics of regulation. So you have transcription factors that are irregulants that are mostly active at the beginning and less at the end, and others that are less active at the beginning of the development and more at the end. And this is the same for the hypoblast. You can see exactly the same graduation of activity. And we do not have so much ones that are really constant over time for these two tissues, totally distinct from the epiblast here. So I will just show you for one factor. After you can do what I'm going to show for all the transcription factors, but it will be too long. So this is HNF1 beta. HNF1 beta is expressed mostly in the hypoblast. And what you can see here is that the motif of HNF1 beta is mostly accessible in the hypoblast here, but in most of the cells of the hypoblast. But when you look at the irregular expressions, you can see that despite the fact that all these cells seems to be able to bind HNF1 beta, the irregulants will only be active at the later stages, as you can see here in green and not here where it is more blue. You can also confirm that this motif is occupied by at least one of these transcription factors by looking at the imprint of the transcription factor, the footprint of the transcription factors at this loci. And it helps you to see that in the hypoblast, something is bound to these factors where it is not no protein are bound in the other tissues. So it confirms the specificity of these transcription factors and probably of its targets. So just to conclude, I think I'm on time. Yes, more or less. So the single-cell RNA-seq alone help us to better identify cell subpopulations and to identify new ones or to confirm some that are well known now. But using the single-cell multiomics, we were able to better refine gene regulatory networks dynamics and how they differ in the way of they regulate their targets in the epiblast and extra-embryonic cells. We can see that there is a rapid shift in these modules of regulation at the onset of elongation, just before gastrulation, while the epiblast maintains a stable pluripotent regulatory states before gastrulation. So now I just want to thank Adrien Dufour, who made all these studies, and people from my team and our collaborators and founders. Thank you very much.
Thanks, Eve. This is very nice work. Other questions? I don't see any in the chat. Maybe I could ask one to get started. So you found epiblast, but Fernando didn't. Do you think it's potentially that the age of the embryo you're looking at is much older and so probably has a lot more epiblast cells, or do you think it's the methods?
Yeah, I can explain this. It's at E5 you have like 100 cells per embryo.
Oh.
So in pigs, when you can get 30 embryos, 20 embryos per throw, you can get enough cells to do 110x genomics library.
Okay.
But when you're working with cows, it's much more difficult. So that's one reason why it's more difficult to get enough cells to do it with bovine embryos.
Okay. But he did say that he could see the cells using other methods, I suppose, some other kind of staining method. So they were there, but
Yeah.
maybe they're just too few.
After you can have bias due to the dissociation procedures, and you can
Yeah.
lose specific cell types. For example, when we have done the multiomics, so we have to prepare nuclei, and we have lost a lot of trophoblastic cells, while we got a lot of hypoblastic cells, and this is a bias due to the technical procedures to purify
Okay.
the nuclei and after to do the libraries.
I was thinking
And
maybe the epiblast cells have fewer RNA, less RNA per cell or something, maybe.
probably because hypoblastic cells are really big ones.
Okay.
Epiblast are really small cells with low small cytoplasm, and probably you have less RNA inside.
So it may be a filtering issue potentially as well.
Yeah.
Okay. Any other questions? Well, that's great. I mean, we're right on time. Any last questions? Okay. Very good. Thanks so much, Eve. And the last speaker for the morning is Praveen from the Research Institute for Farm Animal Biology in Germany. Praveen, are you ready?
Yeah, I'm ready.
Go ahead.
Can I share the screen now?
Yep.
Okay. I think you can see my screen, right?
Yep, it looks good.
Okay. Thank you, Chris, for giving me this opportunity. And today I'm going to give a brief overview of our work related to deconvolution of bulk RNA samples using single cell sequencing samples as a reference in bovine transcriptomics. Okay, coming to deconvolution, it's a approach where we differentiate the different cell types in bulk RNA using computational tools. Basically, it's a cost-effective approach because although single cell sequencing, the cost of single cell sequencing is coming down, it is still comparatively costlier when compared to a bulk RNA sequencing. So when in a scenario where we want to sequence a large number of samples, it is always good to have a set of small number of single cell samples as a reference and bulk samples and to perform deconvolution, which is a cost-effective approach. But the thing is, there are major pitfalls which one has to take care while performing deconvolution because it's not a perfect approach. It has some pitfalls. For example, specifically compared to humans and mice, there is not enough annotations available, cell annotations are available for bovine. So it's comparatively difficult to annotate different cell types in bovine genome. And also when during the experiment, if for example, in some of the cell types, if they are present in bulk RNA but not in single cell, so generally these tools will force the reads which are not present in the single cell into some similar category, which is kind of a noise and bias, which has to be taken care. And also the reference annotation compared to model organisms in the bovine, it's good, but it's not good enough, at least for the single cell to bulk RNA deconvolution analysis. And finally, the technical noise and batch effects, which are quite common. And these, for especially when performed deconvolution analysis, the gene expression signatures in a single cell are considered as directly proportional to bulk samples, but it's not the case all the time. So these things has to be taken care while performing the deconvolution analysis. So here is a data set which we use for our analysis. We have 3 samples, out of which 2 of them already have bulk, corresponding bulk RNA sequencing. And these 3 samples were, we performed the single cell sequencing using 3 different protocols. For the sample 3, we used 2 different protocols, Parse Bioscience and single run. And then after performing this, we executed 2 different approaches to perform deconvolution. One is the normal approach, like using single cell sample as a reference. We perform deconvolution of this bulk, 2 bulk samples. And also for validation, we performed pseudobulk RNA for these 4 sets of these samples, and we performed the deconvolution. So the tools which we use for this analysis are MUSIC, SCDC, SCADEN, and MySort. So all these 4 tools have a different approaches to deconvolute the samples. For example, MUSIC, it's quite popular tool for model organisms, and it can efficiently handle the variability and breed, variability and age of different samples. And also SCDC also uses a similar algorithm like MUSIC, but in addition to that, it uses ensemble weight integration to perform deconvolution analysis. So these 2, MUSIC and SCDC, are kind of similar tools, and the SCADEN and MySort uses machine learning approaches. Coming to SCADEN, it uses deep neural network, and MySort uses signature-based machine learning approach to perform deconvolution. And all these tools have some advantages and disadvantages. I'm not going into details, but we just tested all these tools just to have which one is better for our bovine samples. So here you can see some preliminary results. The plot A shows the actual cell composition of all the 3 samples, and plot B shows the bulk RNA deconvolution cell proportions, which were given by these 4 different tools. And plot C shows the pseudo-bulk RNA deconvolution proportions. But also you can see even in the actual cell proportions for the sample S3, there is a slight variation. And coming to plot B, you can see that MUSIC and SCDC and also MySort are not doing that well. And also SCADEN also able to differentiate all the different cell proportions, but still it's not like the exact results which we are expecting from the actual cell proportions. Same is the case for pseudobulk samples. In SCADEN is able to differentiate at least most of the cell types, but not it's not exactly the exact results which we are expecting. So we thought, for example, if you see here in the plot in the plot B and C, there are a lot of macrophages are macrophage cell proportions, but we thought that it's because there are a lot of cell signatures and features are available for this particular cell type. Because of that, it might be overexpressed. But when we check the number of features, there is no bias exactly. For example, here in Parse Bio samples and also single run, we don't see any major bias. So this is not the reason. So there might be some reason which we should investigate. So based on our preliminary results, we want to have some optimization strategies to further perform the analysis. One is to improve the cell annotation. So we think that the current annotations are not sufficient. We need additional markers and well-expressed markers. And we also want to test more number of deconvolution tools. And also we want to improve the power of our samples. So we want to include more number of samples from public databases. And also we want to perform deconvolution in other tissues and also compare how they are responding and so on. So after optimizing our tools, we want to select 1 or 2 tools, and then we want to develop a scalable and reproducible pipeline using Nextflow. And then after deconvolution, we also want to add an additional layer like identifying CS EQTLs, which are cell-specific EQTLs which can be present in each cell type. So for this, we are exploring different studies, and we found one interesting study, a recent study from Little Paul et al., where it requires input data from expression counts, allele-specific counts, and phase genotypes, and also cell type proportions as an input. And with this, we can find some cell-specific EQTLs. So we are currently working on this approach. So in conclusion, we want to benchmark popular deconvolution tools in the context of bovine tissue samples. So in the future, we are also doing single nucleus sample analysis. So there are some specialized tools like SNUCON and SQUID, etc. So we also want to test these tools, how they are responding. And finally, we want to standardize a deconvolution pipeline with at least 2 tools for bovine tissue sample, and also develop a scalable, reproducible next flow pipeline. And finally, we also want to include CSEQTL analysis to identify regulatory sites at cell resolution. Thank you for your attention. I can take any questions if you have.
Thanks very much, Praveen. Interesting work. We're also working on deconvolution, as you know. Any questions for Praveen?
I do have one. Have you tried the convolution tools to predict cell type transcriptomes? So not cell proportions, but cell type specific transcriptome profiles? Is that
Not yet.
feasible? How well this works?
Yeah, we are still exploring. Maybe it's a good idea. Maybe I will also check how the cell type transcriptomes per se works. Yeah, we have to check this.
Okay. It's something we have in mind as well, but it doesn't seem to be that straightforward.
Yeah, exactly. Yeah, there are many things, many variabilities, even within the samples itself. So, a lot of things to optimize. Yeah.
Fiona, do you want to ask your question?
Thanks.
Or I
Yes,
can.
thanks, Chris.
Go ahead.
Just a quick question with your work. I don't know whether you have looked at the difference between deconvolution accuracy for the nuclei versus the whole cell, single cell results?
Not yet. We are going to do. We already have the samples ready. So in coming months, we are working on the single nuclei, and also want to know how they behave when compared to single cell. We have the liver samples from single nuclei as well. So we want to just compare them and how they behave.
Yeah. Thank you.
Any other questions? Okay, thanks so much, everyone that gave presentations today. I think it was great to have a range of talks on different species, as well as techniques and methods versus straight to the biological questions, and showing the real value of single cell. We have a few minutes where the intent is to discuss a little bit more about what's happening in our single cell task force in a broader sense, and to plan sort of future. And so for those that haven't participated in some of the meetings that we've had, we've met approximately quarterly. And one of the main activities of that is Alex has been organizing these kinds of presentations, longer presentations from different groups in our task force, primarily a bit more focused on developing data and analyzing data. And then there's another activity that's in combination with the Ag Biodata group, which are mostly plant researchers and data scientists. And so we also have meetings with that group, and that group is primarily interested in metadata and data sharing and annotation. And so the next meeting of that group, if you're interested, you can get in touch with me. It'll also be in the recording. And I should mention that again, that we will be posting this recording of the entire hour on the FAANG website, where you should be able to find it easily there in the bulletin board, as well as the task force pages. But the Ag Biodata meeting that's coming up is March 17th. at 10 a.m. U.S. Central Time. And so if you're interested in joining, that's the next meeting. We meet approximately monthly, not every month we meet, but probably 10 out of 12 months per year for the Ag Biodata group. So those were sort of my announcements about what's happening. If you're interested and you haven't joined the Single Cell Task Force, it's quite easy to do. You can select yourself, once you join FAANG, you can select yourself into this task force as well as others. So are there any questions or any comments about what's the activities of the task force that we could discuss? Any
I just
changes?
want,
Go ahead, Alex.
well, I mean, it's not a question, it's just want to remind that within our seminars, regular seminars, monthly seminars, Clarissa Strader will speak the day before on March 16, 5 p.m. Barcelona time, Central European time. The time zones are specified in the emails I have been sending about that. And she will be talking about single cell omics to understand muscle marbling and in cattle and as well in the mouse heart, in the murine heart. That will be in March 16th.
Do we have a list of upcoming talks, Alex, that maybe we could put on the FAANG website? Maybe that would be another place. We're trying to direct people to that as a place for information. So I think as long as we have somebody that's helping to administer that, maybe we can use that as an opportunity.
Perfect. I mean, at the moment, this is the last talk we have organized, and
Okay.
I haven't got any. If anyone has any suggestions or would like to present or think someone would be happy to present, let us know, let me know, because we are fulfilling the list now, and Clarissa is the last in the list
Okay.
for the time being.
We could
I'm
have
happy to
a
share once.
community. I'm sorry, I'm just going to say we could have a community discussion in April. Maybe that would be for next steps, follow up from this session and
Sure. Yeah.
potentially.
Yeah. I'm happy to help organizing this.
Okay. Thanks, Alex. Okay, if there's no other questions, we'll close the session. Thanks everyone for coming. We had 45 to 50 here for the full hour, and I appreciate that. Thanks again to all the speakers, and we'll see you at least next October, I think, is our next session for the entire FAANG group.
Bye-bye.
Bye-bye.
Thank you. Bye, everyone.
you.
Bye.
Bye-bye.